there are a whole bunch of functions, and many of them are rather complicated. I mean, have a complicated set of options. The kind of the first source of trying to figure out about options for one of these functions is to look at the help file associated with it. Say every function in R and in all R packages will have a help file associated with it. Simplest way to get it is to type question mark and then the name of the function you're interested in. Like type question mark read cross. The help file for read cross will show up over there. And in this case, it's rather verbose. I mean, there are a lot of options and a lot of different formats. At the bottom of one of these help files, there'll usually be a set of examples, which might be the first thing that you'd read, is just jump to the bottom and look at what examples there are. Also at the bottom, there's a link to an index for all the help files for that package. It tells you what package that function is in, and if you click index, it'll give you a list of all the functions in RQTL. And it's how you can use this to search for things. Or to, that's probably, I mean, the first source of information would be looking at the help file, and particularly looking at the examples in there. But at this point, this package has a lot of functions, and so it's dull reading to try to go through there and find the ones you're interested in. So the QTL charts package, if you loaded it by typing library QTL charts, and you can type that again if you want. I'm not sure what that error warning message means, but the QTL charts package has interactive versions of a number of these static plots we've looked at. I think in particular, there's an interactive genetic map plot called iPlot map. So it's just like plot map with an I in the front of it. And I can give that my cross object, and it will show up the genetic map over here again. But this time, if I hover over markers, it will tell me the marker name and the centimorgan position of that marker. You can also search for particular markers in the search box at the top, and find a marker that have it highlighted. Yeah.
I assume the I is interactive. Is that what that
yes.
means? Interactive.
Probably one of the more challenging aspects of programming is finding names of things and finding consistent names for things. And I'm not terribly good at that, but this QTL charts package uses a lot of this, what's called camel case, where the name of a function is a bunch of words that are, other than the first one, capitalized.
I just have a question about the R. Why somehow accidentally it go to the plus instead of the red one? How can it go back to the command?
Yeah, if I start a command and it goes to this, for me, if I type escape, it'll stop. It might be controls. For me, it's escape.
Escape.
There's also a stop button at the top of the console that you can use if you get stuck.
find that error messages are nicer if transmitted by a cat. So if a cat is telling you the error that you made, then it just is not quite as painful. Yeah, so that, I mean, that's sort of the basics of getting the data in and looking at basic aspects of it. But we haven't yet gotten to QTL mapping. We're going to go back to the slides and talk a little bit more. So that, I mean, we have our data are, we have some experimental cross, say a back cross or an inner cross. We have a bunch of individuals. We have phenotypes for all of them, some quantitative phenotypes. We have genetic markers across the genome. We know where they are on chromosomes, and we have, for every individual in an inner cross, which of the 3 genotypes it has, or it could be missing. The simplest way to analyze this data to try to find QTL is to simply look at each marker one at a time and say, and then split the individuals according to their genotype at that marker. So in a back cross, there'll be just 2 groups. In an inner cross, there'll be 3 groups. And then do a statistical test to say, are there differences in the average phenotype across those 2 or 3 groups? So in an inner cross, you split the individuals into the 3 groups at a marker, and then do analysis of variance to say, are these 3 averages, do they look different or not? And we look for markers like this where there's a pretty big difference between the 3 groups. And we are less interested in markers like this where the 3 groups are pretty much the same average phenotype. And we just repeat that for every marker. The advantages of this are that it's simple. You can easily incorporate covariates if you want. Like if you want to account for, if you want to account for environments, that you can just, instead of doing a t-test, you can do a linear regression. You can easily extend this to more complicated models because you're just, the genotype at the marker genotype is just one covariate in some larger model that you might have. So if you want to fit your, full fancy mixed effects model incorporating all kinds of block and plot effects, you can do that and just shove a marker in there in the place of a QTL. And you can start doing this before you have a genetic map. Those of you that are working on an organism where you don't have a genetic map, once you have the marker data, you can just start looking, and if you have genotypes and phenotypes, you can start looking for associations even before you build the genetic map. And I recommend doing that because the genetic map may take you 3 months, and it's good to have some sense that some of your markers are actually associated with your phenotypes, up front, even if you have no idea where they are in the genome. The disadvantages of this is that individuals that are missing their genotype at that marker, you don't know which group to put them in, and so you have to just throw them out. And you get imperfect information about locations of QTL because you're just ending up with sort of a bunch of p-values from tests at each marker. And if you have widely spaced markers, you're potentially losing information that you can do better to learn about positions in between markers where you think the QTL actually sit. In practice, really, this is probably the only, for more modern data where we have lots of markers, it's the missing data at a given marker and having to omit those individuals. That's really the main disadvantage that we want to deal with. But probably the biggest disadvantage is that you're really thinking about one QTL at a time. Yeah.
Will we be able to get a copy of your notes later?
Yes. Sorry I didn't mention that. If you go to my website, qtl.org, or kbroman.org, under teaching, a little way down, you'll find today's workshop. So I have these slides are there, and there's also a black and white 200-page version if you want to print it out.
Oh, no, I haven't.
There's also a PDF of the basic tutorial that I'm giving, but I'm not going to follow this exactly because you may want me to do different things. And so most of what I will cover, not including the QTL chart stuff, the interactive graphs, will be in that. So the biggest disadvantage of this, just look at each marker one at a time, is that you're just looking at each marker one at a time. You're just thinking of, you're really imagining that there's one locus that's affecting the trait. You're not if you find on one chromosome there's a really big effect, when you look at another chromosome, you're not taking that into account, and so you're losing out of, you could do better. The principal disadvantage of just looking at the markers one at a time is having to omit individuals that are missing genotype. That the main solution to that problem is this method called interval mapping. That the landmark paper in QTL analysis is this Lander and Botstein paper in 1989, which if you do QTL analysis at some point in your life, you should try to read it. Although it is maybe not the best source to start learning QTL analysis. It's worth looking at. It kind of covers all essential issues back there in 1989. What interval mapping does is it's like doing an analysis of variance at a marker. It assumes that there's one and only one QTL in the genome, but then considers each position one at a time. Each position individually in the genome as the location of that QTL. Some arbitrary location. But that location need not be at a marker, but could be at any position in between markers. And imagine that we knew QTL genotype for an individual at a marker. They assume a model that's really the same model as if you did analysis of variance at a marker, which is that the phenotype of an individual, given its QTL genotype, follows a normal distribution with some mean, depending on the genotype, and some common SD. So, imagining that in a backcross, there's the individuals that are CC active QTL have some normal distribution, and the individuals that are heterozygous have the same normal distribution shifted. And in intercross, the other homozygote, they have basically the same normal distribution shifted over yet again. So we're imagining there's one and only QTL, and if I told you everyone's genotype at that QTL, then the phenotypes of those individuals would follow three normal distributions that should look exactly the same, but just shifted relative to each other. That's exactly the model that you use in analysis of variance, where you split the mice into three groups and do analysis of variance. Here, the problem is we don't know anybody's genotype at the QTL. But what we can do is use the information about all the surrounding markers to calculate the probability that an individual has is in this group versus this group versus that group. So, thinking in a back cross where on that recombinant chromosome you have one of two alleles. Here I'm going to say A or B, and say I have a given individual that has an A allele at the first 5 markers and a B allele at the last one. And what we want to know is, imagining that the QTL is sitting there. We don't know its genotype at that position, but we can calculate the chance that it has either an A or a B at that location. The simplest model is, the simplest assumption to make is that these genotypes are without error. There's no possibility that's the true genotype at those locations, and that the recombination process exhibits no crossover interference, which means the locations of these crossover events are at random. At random in a certain way. Whether there's a crossover in one interval is independent of whether there's a crossover in an adjacent interval. With that assumption, in trying to say what's the chance that that individual has either an A or a B at that location, we just need to look at the 2 nearest flanking markers. And then the question, is there an A here or a B here, is really, well, we did not see a recombination event across that larger interval. What's the chance that there was a crossover both here and here, given that it didn't recombine? So, if we need to know the recombination fraction for that interval and the recombination fraction for that interval, and then we do a little calculation to figure out what's the chance it's an A versus a B there. Yeah. We look at each individual one at a time. Because each individual is independent.
So you already have the
Right. We need to have estimates of those recombination frequencies prior to doing this calculation. So if a different individual, with different genotypes, if a different individual has a B here, A, missing the genotype here, and a B here, and an A, if we assume no genotyping error and no crossover interference, then we just need to look at, for that individual, the 2 nearest flanking typed markers. So we look at this marker and that one, and ask, given it's A and then B, what's the chance that it's A here? Yeah.
Given the fact that we're recording this and we're recording the scientific questions that you guys are asking, if you ask a question, will you make sure to say it loud enough so that it gets on the recording? But if for some reason you don't, if you don't mind repeating it, because I don't even know what the question was.
Oh, I'm sorry.
No, that's okay. I just figured this was the first time somebody really asked a true scientific question, so we might as well start now.
Thank you. I will attempt to repeat questions
Okay.
as best I can.
Okay.
Focusing particularly on the rest of you in the room, but also for posterity. The question was, do you need to consider all individuals jointly, or can you, and my answer was, in these kinds of crosses, that individuals are independent, so we can just work with one individual at a time. So, for a given QTL location, I look at each individual one at a time, and I say, what are the 2 nearest flanking typed markers? And then, based on the genotypes of the 2 nearest flanking markers for that individual, do a calculation of is it A or B at that location. For this individual, I know that there was a recombination event across that broader interval. And the question is, did it happen to the left of the QTL, or did it happen to the right of the QTL? And that ends up being pretty close to, the relative length of that to that, in a way. This interval is a little bit longer than this one, so it should be somewhat less than a half of being B.
somewhat more likely to be like the marker that it's closest to. So this individual, again, I mean, so A, B, missing B, A, I can, if I assume no genotyping error, no crossover interference, I just look at the two nearest blanking type markers and I ask, was there, given that there was no recombination event in the broader interval, what is the chance that there was a double crossover versus not? The no crossover interference assumption is a very strong assumption. And of all the approximations we make in QTL mapping, it's maybe the most blatantly false, like far from the truth assumption. If you look at this data, I have a crossover here and a crossover here. Recombination events, both the intervals around this. The chance of, I maybe need to know about distances on this chromosome, but the chance of there being 4 crossovers in a row here is really small. And that there's a crossover here and here really should influence my understanding of I should say there's a greater chance that there's not a recombination event. I mean, there's not a crossover in that interval. I should say it's more likely to be just B. Thanks.
That's within a single generation, right? Over real. These are real. They are some.
That's right. The pattern of crossing over along a recombinant inbred line's chromosome is closer to no crossover interference, even if there's very strong crossover interference at each generation of meiosis. I was supposed to repeat the question, wasn't I? Okay. So this assumption is blatantly false, but is hugely convenient for all these calculations because we just need to, it makes all the calculations about these kinds of genotype reconstructions really easy. And the effects of, efforts to incorporate crossover interference are a huge pain and turn out to not have any influence on the results at all, really. I would like there to be some effect of crossover interference on the calculations, partly because I'm really interested in crossover interference, and I would be able to write a whole bunch of papers about how to account for crossover interference in these calculations, but it doesn't really matter, and this is way easier. So in practice, we don't actually do the calculations with these, this assumption is seldom true. The lack of genotyping errors is maybe as far from the truth as this one. But just the calculations I've explained turn out to be kind of a pain to write in code. And what we actually use is what's called a hidden Markov model that allows for genotyping errors and can incorporate markers that are strictly dominant. And just the calculations are easier. And what we do in practice is form like a grid of putative QTL positions along the chromosome, and then all at once calculate the genotype probabilities at every one of those positions. But the essence of it, I think, to think about is, imagine that there were no genotyping errors and no crossover interference, that you're just really looking at, nearby markers and making inference about, or, using that to calculate the probability that an individual has that, what possible a probability for each of the possible QTL genotypes. Having done that, or that's sort of the first part of this interval mapping method. The next thing is using that to actually do the calculation about to do a test of association between genotype and phenotype where you don't know anybody's genotype at the QTL position, you just know the probability of their genotypes. A useful sort of simple case is to think of backcross, where each individual has one of two genotypes, and just a pair of markers surrounding one QTL. And suppose all the individuals, their genotype at this marker, their genotype at this marker. So then there are really just four types of individuals. There are those that are heterozygous at both markers. There are those that are homozygous at both markers. There are those that are heterozygous at the first, homozygous at the second, homozygous at the first, heterozygous at the second. The individuals that are heterozygous at both markers are almost all heterozygous at the QTL. The closer the two markers are, then the more likely that all those individuals are also heterozygous at the QTL. So the phenotype distribution for these individuals will just follow a normal distribution centered at the average phenotype for the heterozygotes at the QTL. The individuals that are homozygous at both markers, their phenotype distribution will be normally distributed around some other mean. Those that are heterozygous at the first and homozygous at the second, they will include two groups of individuals. You don't know which one's which, but some of the individuals will actually be heterozygous at the QTL, and some will be homozygous at the QTL. So those that have a recombination event in the second interval, so that their QTL genotype is the same as the first marker, and those that had a recombination event in the first interval, so their QTL genotype is the same as the second marker. So this group of individuals, there'll be some that are heterozygous at the QTL, their phenotype will follow this normal distribution, and some that are homozygous at the QTL, their phenotype will follow that normal distribution. Altogether, they follow what's called a mixture of those two normal distributions. Just basically add those two normal distributions up, weighted by the relative proportions of the two groups. This group of animals looks like the opposite of this. A smaller proportion that are heterozygous at the QTL, a larger proportion that are homozygous at the QTL, and overall, follow that mixture. What our data will be, we'll know who these four groups of animals are, and we'll have like a histogram of the phenotypes within those four groups. And really what we want, but we won't know these underlying distributions, but we can use what's going on here and a little bit about them to estimate those two underlying averages. And that's what interval mapping does. Is it uses the same model as if you just did analysis of variance at a marker. But instead of knowing everyone's genotype, you just know the probabilities of the two possible genotypes, or the three possible genotypes in an intercross. And you need to combine that with their phenotypes to figure out what the underlying averages are.